when you have transcriptomics data for a particular disease, for instance, glioblastoma, you can actually start analyzing it, and then you're able to get such nice network like here, which describe the molecular mechanism of the disease. You can also see the central point of that mechanism. And in the end, you can even identify the important drugs which can be applied for this disease in priority with a drug score associated. You can find new drugs which may be repurposed for this disease. And all this can be done with Genome Enhancer, which you see here. But let's start from the very beginning. Once you have data, like here, for instance, this is a data about glioblastoma patients, which are, transcriptomics was profiled for each of the patient. For instance, this one patient, samples from it was done by transcriptomics. So you can get this data from this omnibus entry, basically copy, and now go to the Genome Enhancer, create the new entry, new project, and put this URL here to upload the data. So you see the data is uploading now into the platform. And once it's uploaded, you click on import, and the data are imported. First, you have to set this is transcriptomics, of course, and after it's imported inside the platform, it's ready to analyze. So first you copy all the data in this gray box, which you can now spread over the experiment and control. You put all the patient data into experiment box and all the control data into the control box, and proceed further. You select the disease you're analyzing, so glioblastoma, or you extend it to the wider category, glioma, and then you select the experiment versus control and start the analysis. It takes maybe a couple of hours, but then you get the full report here, which tells you which are the most important master regulators or druggable targets and which transcription factors found to be most important. You see this data. You can also see which differentially expressed genes were found and how they were, Heatmap for them. You also see, of course, how these genes are regulating in the pathways like this axon guidance, but most importantly, analysis of promoters of the differentially expressed genes are done, and then transcription factors which are clustered in the promoters of those genes are identified. As you can see on this map, and these are the promoters of upregulated genes, and you see the clusters of the binding sites identified as the most important drivers. And at the end, you get, of course, this nice network which explains the whole mechanism of the disease control. You see here the differentially expressed genes and the master regulators like here CD44, MMP, and also new master regulators like VEGF, which are maybe not very characteristic for glioblastoma, but here they were clearly identified. And you see also transcription factors which are activated in this network. Also, you can identify those most important druggable targets and the core of the mechanism, which explains you which are the most important master regulators. And at the end, you get the drugs which are known for glioblastoma, which are used, but they are prioritized. And then you see those drugs which may be repurposed here and which are under clinical trials at the moment. You can also get those active compounds like curcumin, which might be useful here. All the anti-cancer drugs are then prioritized here by giving them a respective score, drug score. And at the end, you get the conclusion of that analysis and all the disclaimers and the method sections. So with that, you can see how it is easy to analyze your gene expression data. You don't need to ask and wait for bioinformaticians to analyze your data. You just drag and drop it in the box and get the results. Thank you. If you want to try how our tools work, register for this free geneXplain platform. Below is the link. There, you can upload your data and apply more than 200 different tools and workflows. If you want to learn more about molecular mechanism of glioblastoma, click the link in the video description and read our blog article. You will discover more than 1,000 of biomarkers of glioblastoma, which are classified into 7 different subtypes, such as causal biomarkers, correlative and prognostic biomarkers, and many others, such as, for instance, therapeutic targets and biomarkers associated with the disease mechanism.